Hello, my name is Sabrina Templo. This video is for Bio 225, Week 7. Today is June 24th, 2020, and the experiment today is called Antibiotic Sensitivity Kirby Bauer Diffusion Test. In this experiment, I will be performing a Kirby Bauer diffusion test with Staphylococcus epidermidis and 3 antibiotics, penicillin, novobiocin, and gentamicin, which I already have over here. I've cleaned my surface with 10% bleach solution, and then I have all my materials listed in for this experiment. Next, I already used a permanent marker, and I already poured my agar plate yesterday to prepare for this experiment. With a permanent marker, I created 3 sections, which is labeled with the corresponding antibiotic. So this is going to be NVB, which stands for novobiocin. PNC is for penicillin, and GNT is for gentamicin. So first, what I'm going to do is flip the plate over and have it a little bit open. And then I'm going to have my pipette. It's been sitting in alcohol. I'm gonna make sure that it's dry. Then I'm gonna collect from my active culture broth Staphylococcus epidermidis. But first, I'm gonna sterilize the lip by passing over the flame. Then I'm gonna collect with my pipette, 0.5 ml. And then I'm gonna transfer 8 drops to my agar plate. Then I'm gonna dispose of this in my bleach. And then I'm gonna reflame my active culture. And then I'm gonna place it back in the refrigerator for future experiments. And then in the lab kit, I've been provided with an L-shaped spreader. So when I open it, I'm only gonna touch the long straight end. And it's already sterilized because it's in sterilized packaging. And I'm gonna use it to spread the broth around the agar plate evenly. Doing this so the bacteria gets all around the agar plate. Then I'm gonna place this in the bleach. Now I'm gonna allow my agar plate to sit undisturbed with lid side up approximately for about 1 to 2 hours until the broth is absorbed by the agar. Then I'll perform the rest of the experiment. Hello, this is a continuation of my experiment antibiotic sensitivity Kirby Bauer's diffusion test. I allowed my agar plate to remain undisturbed, lid side up, for about 2 hours now, until the broth has been fully absorbed into my agar. And now I already placed the antibiotics in the plate. I've done 2 of the antibiotics. The first disc was the gentamicin, which is this one. I already placed in the agar. And I also did the novobiocin right over here. And I will demonstrate how to do the penicillin. So I have my tweezers that have been sitting in the alcohol for about 30 seconds from doing the previous antibiotic. And I'm gonna go ahead and shake it dry. Then I will be taking my antibiotic disc of penicillin. And with the tweezers, I'm going to go ahead and collect the disc. So now with the disc, I'm going to transfer it to the plate. I'm going to place a disc on the plate in the section that is labeled PNC, penicillin. Then gently with the tweezers, press it down a little bit with a little bit of pressure into the agar so it is secure. So now with this, I will let the plate remain lid side up for about 10 minutes so that the disc can secure and place in the agar surface. And then after 10 minutes, I will invert the plate, which I will have the bottom side, agar side up, and put it in the incubation location for 12 hours. My hypothesis for this experiment is that the Staphylococcus epidermidis, being a gram-positive bacteria, will exhibit resistance to novobiusin and gentamicin and will be inhibited by penicillin because it is a bacterial narrow spectrum that targets gram-positive bacteria, and the bacteria cells will lead to its death. So after 12 hours, I will check the diffusion plate for bacterial growth, which will appear as a white field, which will cover the agar, and then it will look like somewhat like this photo. Here you can see the inhibition zone right over here, where there is no growth, no bacterial growth. And that's what I will be measuring with the ruler right over here from my lab kit. And that will be the measuring of the inhibition zone and the resistance to the antibiotic. So after the 12 hours, I will do the observation. Thank you.